Hello and welcome back. So we've talked a little bit about mitosis. Actually, we've talked a lot about mitosis, and we know that that is how cells reproduce. And today we want to talk about reproduction in general. What is the outcome of all of this? And we know about growth and repair and, development. But when we start looking at the grand scheme of things, how do we continue life? And that is through reproduction. And there are a couple of ways that that happens. The oldest of the forms of reproduction, and probably the simplest, is asexual reproduction, and that is where mitosis comes in. So in this case, you have one cell, and it's going to become more cells, right? So this is what we did for about the first 2 billion years. Started out with all your little single cell guys. Sexual reproduction starts out with single cell guys too, but that comes later, right? So first 2 billion years, we're just simply cloning, dividing. What are the advantages of that? Well, it's quick. You don't have to look for a mate, right? You just divide. That's great. Quick, easy. You can increase your population very rapidly, right? So you can get huge numbers of offspring because it's an exponential growth, right? 1 to 2 to 4, etc. Now, this is really favorable in a stable environment, but it is problematic if the environment is going to change. So, that brings us to the disadvantages. The offspring, with the exception of mutations, right, mistakes in mitosis, mutation or, right, and or, mistakes in mitosis, the offspring should be identical. They really should be clones, okay? There's no variety. What this means is that you have less diversity over time, evolution is very slow, and asexual reproducers are more prone to extinction in changing environments. In stable, it's great. In changing, not so much. Okay, so let's talk a little bit about the types of asexual reproduction. We've got fission. This is where you have one adult that splits into two. Okay, I guess they're all adults if you look at it that way, but you have one organism that splits into two. You can see this guy's dividing right here. This is a planaria. Then you have an anemone that's dividing. You have some coral action over here that's dividing. It's very exciting. Now, a slightly different take on fission is budding. And in this case, you have a group of cells on a parent, right? And this is Hydra, on a parent that develops into a new individual. So this little cluster of cells started dividing to become a new little thing, a bud, that will drop off. And this, you can do this as an internal or external thing. I'm not going to ask you about that. I just want you to know that this is where, instead of the individual dividing, you have a small cluster of cells that makes a new one. And then there's fragmentation, which it can be reproduction. Like if you cut an earthworm in the right place, you can get 2 earthworms. Starfish, you can cut those up and get more starfish. Although fragmentation mostly is about regenerating body parts, it can regenerate individuals. Now, another form of asexual reproduction that you may be less familiar with is parthenogenesis. And this is where you actually have organisms who lay eggs, right? And they hatch, and that's all very exciting. It's called, you can think of it as kind of like a virgin birth, right? They're all females. The eggs develop without fertilization, okay? Because the eggs are clones of the mother, all right? And so you have the same idea. You have this egg, right? But it's all already diploid. It doesn't have to be fertilized. So the only difference between the actual development process in parthenogenesis and sexual reproduction is that you start out with a diploid cell that is a clone of the mother, okay? And then everything else progresses normally after that. Which brings us to sexual reproduction, to become one, right? And so in this case, you've got to deal with some specialized cells. So first I just want to mention that we have 46 chromosomes. As you probably know, this is probably a review. We'll talk about this more later on. But we have 23 pairs, okay? And these are all the same, right? These are all just, this chromosome 1, chromosome 2. You have 2 copies, one from mom, one from dad, okay? But this last one, this 23rd pair, is special. This is the sex pair, okay? And what you're looking at here is an X and an X, which means this is a female. But here's how it relates to what we're talking about. In order to get this matched set, you have to have 2 single sets come together, Okay, and that now we are talking about eggs and sperm, right? So we want to do a couple of things here. We want to talk a little bit about what gametes are, right? What they mean, and then we want to talk about how gametes are made, and we want to talk about later on, not today, how gametes fuse and meet and fuse to become a diploid cell, right? Diploid, this is a diploid chromosome set. It means that you have two copies of each chromosome. So we're going to talk about the egg. We'll talk about sperm a little bit later on, but I just want to talk about eggs here today because eggs are cool. This is the gamete. Gametes are sex cells. This is the gamete produced by the female. They are haploid. Sperm are also haploid. That means that they have one set of chromosomes. Okay. Now the egg is cool because it provides everything that you need to have to make the zygote, the fertilized cell, except the other half of the DNA, which comes from the sperm. So the sperm provides only half of the DNA. Everything else comes from the egg, and that's why eggs are huge, right? And I just want to take a moment to talk about the varied world of eggs. So eggs can come in all sorts of shapes and sizes, and some of them are really cool, okay? And so I've just put some in here. This is a butterfly egg. This is a different butterfly egg. But you can see all these great structures, and they're all protective structures. And then this is a praying mantis egg case, and there are anywhere between 1,000 and 1,500 individual praying mantis eggs inside of this case that's stuck to the tree. So there's some insect eggs. Here's some fish eggs. I love these because you can actually see little baby fish inside. Really cute. Okay. And then here are some sharks. Sharks, I think, have some of the most beautiful and unique egg cases. And sometimes they hang like little purses, right? And here's a little shark developing inside. It's very cool. And then here are some amphibians. And these are neat because, again, you've got these clear cell membranes, and so you can actually see this is a cell that's divided. This down here is going to become the yolk, and this up here is going to become the frog, right? And then here you have this little like spit bubble nest. Here they're all clustered underwater, and then tree frogs actually glue them to leaves. Really fun. Reptiles, now you can see a transition between that water membrane. You're now moving into terrestrial world, so you need special things to live in a terrestrial environment. And the first one, and we're going to talk about these throughout the class, right? But the big one is that you need to have an egg that is permeable to gases but won't lose the fluids inside and won't allow fluids from the outside to come in. And so you get this egg, this leathery kind of shell that you see on reptiles, which then, oh, these are just adorable. I mean, how adorable are turtles, baby turtles and baby lizards? Super cute. Now, this becomes more calcified when you start talking about birds. And birds, they're all the same shape because they all have to come out of the bird the same way. And that's kind of true for reptile eggs. They're all coming out the same way. So they're all a little bit different. But I mean, they're all similar shaped, but it's the colors and patterns. And birds' eggs have some of the most amazing patterns and colors. And these are actually probably in your grocery store right here. So check that out at some point. And then, of course, are two mammals that also lay eggs. They are the monotremes. And again, we'll talk about them later when we talk about mammals. They're the only mammals that lay eggs. And here is a platypus egg, right? And they're little tiny platypus babies. Maybe cute, maybe not. You be the judge. And here is an echidna egg, and there's that little echidna popping right out. Also, maybe cute, maybe not. You be the judge. Okay. So, how do we make eggs? How do we make sperm? So, if you want to just faithfully reproduce a cell, that is mitosis. If you're trying to make gametes, that is meiosis. And I love this because it's a false color image of the DNA. Well, it's not a false color image. It's a tagged image. This is fluorescent tags in the DNA. And so you can actually see it's very cool. And you can go from this tangled chromatin to this is the nucleus of the egg, right? Which is pretty neat. So how does that work? Meiosis, unlike mitosis, has two phases. There's meiosis 1 and meiosis 2. These are very similar to mitosis. Okay, the same things are happening in prophase, metaphase, anaphase, and telophase as in mitosis. Okay, there are a couple of significant differences. One, the big one, is that you do it twice, right? And then the other one I'm going to talk about in just a second. So we start out the same way in prophase. We've got the DNA condensing into the chromosomes, all that good stuff. In metaphase, oh, here you're attaching your pairs. In metaphase, you're lining up on the equatorial plate. In anaphase, you're pulling the chromosomes apart. And then in telophase, you're going to have that split, okay? Just like in mitosis, right? Now, chromosomes are single pieces of DNA that have been coiled 4 different times, right? And so you get this dense structure. So it's a coil, then a coil, then a coil. They're coiled around proteins, right? Remember I told you proteins do lots of different things, but there's little protein balls, and the DNA actually coils around those balls, and then those balls coil up, and those are called histones. Okay, so this is just the same that I showed you before. Okay, so you have an idea of what's going on here, what you're actually looking at. Okay, so what happened? So here is your next question. Which of the following is not a way that sexual reproduction introduces variety? So, right, if we look at all of these, when we say faithfully reproducing each parental diploid cell, is that mitosis or meiosis? That is mitosis. Now, producing gametes with different combinations of parental chromosomes, that's meiosis. Allowing recombination of alleles, we're going to talk about that in just a second, that's meiosis. And allowing the combination of chromosomes from 2 different individuals, that's meiosis. So these are the 3 ways that sexual reproduction introduces variety, and we're going to talk about all of these a lot more throughout the class. So let's first start off with this idea of homologous chromosomes. We know that, say in humans, we've got 23 pairs, okay? What that means is that you've got 1 and 2. So these 2 together, this orange one and this blue one, would make a pair of chromosomes. Then when they're duplicated, right, here you have the cute little duplicated structure, right? So now you've got the original and the replicant, if you will, right? Daughter, okay? Now, we're going to talk later on when we talk about genetics. Each person has 2 chromosomes. So there's a 50-50 chance that either of those chromosomes will end up in the egg. And so eggs or sperm can have different, right, alleles. Each of the half of the chromosome pair would be an allele, okay? Have different alleles of the genes, right? And we'll talk about this more later on, okay? So genes are locations on each of the chromosomes that will code for various proteins that give you various traits, okay? Some other things too, but that's what we'll be talking about. And so each of those are called alleles. So if, let's say that you have 1 allele on this one and 1 allele on this one, and they're for the same gene but they're different versions, you have a 50-50 shot of either of these genes, or either of these versions of the gene ending up in the egg or sperm, and that's pretty cool. Again, we're going to talk about this part more later on, so if you're a little bit confused, that's okay, don't worry about it. We're going to talk about it a little bit more later on. But what we are going to talk about right now is the idea of recombination. So one of the other ways is that you actually have a change in the order of genes on the chromosomes, because remember, this chromosome is a little bit different than this chromosome. Even though they are a pair together, they are a little bit different. Sometimes they will cross over, right? This is called crossing over, and their little legs will break off and reform. So now you have the original genes from this chromosome, but it's joined with the original genes from this chromosome, and so you're going to actually have a different combination of genes. And again, we'll look at the details of this later, okay? But crossing over sort of jumbles up the genes, right? And so you get a different variety, yeah? And then here's just an example. Let's say that you have this chromosome, we've got little a gene, big A gene. So they're A's, they're the same. Little b, big B, right? They're the same, okay? Big C, little c. When you get crossing over, you can see now that your big C, instead of being with your big A and big B, is now with a little a and little b. And that's how you get combination. So these are faithful. These are the same as the parents, okay? And these are the recombinants. They are a different, right, combination than the parents, okay? All right, now crossing over happens in meiosis 1. Okay, crossing over happens in meiosis 1. That is going to be during prophase. It's very exciting. So, by the time you get to meiosis 2, this is the second division, you have this sort of telophase that's happened, right? It's kind of divided. But you see that you have 2 diploid nuclei, right? And so if this were mitosis, we'd be done, yeah? But it's not. So what we're going to do is we're going to go into metaphase again, right? And this time, instead of pulling the 2 pairs of chromosomes apart, we're going to have the homologous chromosomes pulled apart, so that in the resulting cells in anaphase, right, you have a single chromosome instead of a chromosome pair that is going to end up in the new cell. And so in telophase, instead of getting 2 diploid cells, right, you get 4 haploid cells. Okay, so just to review really quickly, you have prophase, right, and this is where you have the condensation and the crossing over. In metaphase 1, the chromosome pairs, because they were replicated earlier, the chromosome pairs, right, all line up. In anaphase, they're pulled apart, so you have 2 diploid regions, okay? In telophase 1, you get a contractile ring and it starts to split, okay? And then prophase 2 starts, right? And then you have metaphase 2, and in this case, you're going to have the chromosomes line up, right? And then they're going to be split in half. So by telophase, you have 1 chromosome in each of the new resulting cells at the end of mitosis 2. All right, so what are the benefits of all this? And don't worry, we're going to go through this process in class. You're going to do an activity where you actually do mitosis and meiosis. Well, you've already done mitosis, but you should do meiosis. All right, so the benefits and costs. Benefits. Recombination is wonderful because it gives you all this variety. And this is great because the offspring are a little bit, or a lot, different than the parents, and so this may actually make the offspring better suited to a certain environment. This is great in changing conditions. It may also provide protection against various diseases. If you're a clone, everybody's susceptible to the same diseases, but with this variety, some are more susceptible and some are less susceptible, and that's great. Okay. Now, there are disadvantages, and this is sometimes called the cost of males, right? An individual transmits only half of its genome to the next generation. So the idea is that it's the goal of the male is to have as many offspring as possible, to have its genes in as many offspring as possible. The goal of the female is to have all of her offspring survive, right? Or as many of her offspring survive as possible. That's also a goal of males, but it usually falls to the females to make sure that happens, which is the double cost of females, right? The double cost of females is that she's only giving half, right? The typical cost of males, but she's also sacrificing fitness for the health of the offspring, okay? And so all of this adds up to a growth rate of populations that's slower, right? You have to find mates, you have to actually mate, the offspring have to grow, right? Then those offspring also have to find mates, and then they have to grow up to sexual maturity, and so on, okay? And so that's the disadvantage there. Now, we have 2 different sexes in lots of systems. In birds, you have a ZY system. In fruit flies, you have an X and, XO system. But we're going to talk about mammals. In mammals, you have an XY system, and the male determines the sex in the offspring. In birds, by the way, it's the female. But in humans, mammals, it's the male. So what that means is that males have an XY, and females have an XX. But it turns out that the number of chromosomes that you have is directly proportional to the amount of proteins that will be made from those genes. So what we're saying here is that in the XY system, the X, right, in the male is going to produce a certain amount of a gene. But females have two X's, so they're going to produce twice as much of that protein. And that seems like that would be bad. And in many cases it is bad because you don't want too little, you don't want too much. So how does that get dealt with? Okay? And it turns out that you get something called X inactivation. Okay, and X inactivation is responsible for calico cats. All of these kitties, all of your calico kitties are girls, right? So let's say that you have your zygote and you have two variations of a gene. So in this case, you have the orange allele and you have the black allele. And remember, we'll be talking about alleles more later on, okay? Well, in this case, in the males, you would have either the orange or the black. So what happens is one of the X's, for all things, right? One of the X's is inactivated. It is condensed and covered up with methyl groups, so none of the DNA can be read, and that forms what's called a bar body, right? And let me get some more pictures up here, okay? And this happens sometime during early development. But in some animals it happens a little bit earlier. In some individuals it happens a little bit later. The earlier the X inactivation occurs, the larger the patches of color you'll get, right? So in this little guy, it was very early in development because here, right, this bar body was orange. So all of the daughters of this X are going to be black. All of the daughters of this cell with this X are going to be orange, right? So if you start early, this is going to produce a lot of cells. If you start later, each cell will produce fewer cells because there are already more, okay? So this one happened really late in development, okay? But you can kind of see comparison. And sometimes it happens really early, and sometimes it doesn't happen entirely all the same, right? So, you know, we have this symmetry, and we'll talk about that more later on. Here you have what's called a chimera, right? And so you had some different things going, but this is pretty cool, right? And you can actually see the symmetry, and these cells developed here, and these cells developed on this side, so you have totally different situation. But a lot of this has to do with the X inactivation situation. Now, some species are what we call hermaphrodites, and hermaphrodite is a biological term that describes individuals, and these are going to be actually whole species. All of the individuals in the species have this, but they have both male and female functional characteristics. Some species, like little sea slug here, right here, actually are male and female at the same time. Now they have mechanisms to prevent them from fertilizing themselves, so when they meet their mate, they're both trying to be male, because think about the cost of females having to raise the offspring and put that energy into it. That was a big deal, right? And the more dominant will be the male, end up being the male, and the less dominant will end up being the female, so winner is a male. Which could be argued that maybe they're not the winner, who knows, whatever. Anyway, that's a totally different class. So you have these both, male and female, at the same time. Now some of these species are sequential. They will start out as one sex, and then they will be the opposite sex during a reproductive phase, okay? If they are protogynous, it means they're female first. If they're protandrous, it means that they are male first, okay? We're not going to spend a lot of time talking about this because this does not describe humans, right? We are not a hermaphroditic species. All right, now hybrids. Speaking of species, can you have one species breeding with another species, okay? Are all eggs and sperm compatible? Well, part of that depends on chromosome number, right? Chromosome number is going to vary among species. Chimps have 48, camels have 70, alligators 32. You can read the rest of that. We have 46. So it's very important that the chromosomes match, at least in animals. Plants are way more flexible about this, but in animals, the chromosome number really needs to match. Otherwise, bad things, okay? If you don't have the right number of chromosomes, bad things, okay? Now, just as a review, if a liver cell of an animal has 90 chromosomes, the sperm cell would have how many chromosomes? Twice the diploid number, half the diploid number, half the haploid number, 180, 90, or 45. Well, all of the other cells in the body are diploid, okay? Sperm and egg are haploid. So if you have a diploid cell that's 90, the haploid cell, the sperm cell, would be 45. So the answer is 45. That's just a little bit of a review, okay? Now, chromosome number is really important. If the chromosome number does not match up, okay? Some of the big things that happen right up front are going to be reduced hybrid viability. Now, by the way, this is if those two organisms can even breed together, right? You may have it so it doesn't even work. They may come into breeding season at different times. Their habitats could be isolated. They are not attracted to each other, so there's no mate selection there. It just does not work out, right, physically. Or the gametes don't fuse. They just don't attract. So those are pre-zygotic berries. But let's say that you actually do have the egg and the sperm get together post-zygotic. Okay, we'll talk about zygotes more next time. You can actually have an offspring form. Now, a lot of times what happens is that there's no offspring at all, right? It does not happen, okay? Even if the gametes fuse. But if you do get something, then you have your reduced hybrid viability. So that means that the hybrids fail to develop or to reach sexual maturity. You have reduced hybrid fertility. That means that the hybrids are sterile. Okay, like in the case of mules, and we'll talk about mules later too. And then you have hybrid breakdown. The offspring are sterile. They can have their own offspring, okay, but those have reduced viability or fertility. And so it's very important for animals that chromosome number lines up, and it's why we don't have dog cats, right? It just doesn't work, okay? And some later on we'll talk about, are there humanzees or things like that? Is that possible? But when we start thinking about chromosome numbers, I want you to start thinking about, would that work? Think about how many chromosomes chimps have. Think about how many chromosomes humans have. Okay, so that is it. This one was a little bit longer. We had a lot of information. If you have any questions at all, and I sort of imagine that you probably do, please make sure that you have written them down and that you are going to ask them in class when I see you again. All right, thank you for listening and have a great day.